So I will be sharing with you today the story that we have, obviously developed and now all of you are aware of as well. I'm going to cover kind of the history of our research, looking at runoff impacts on salmon, our discovery of 6-PPD-quinone, and scattered through there some of the research that we're working on still today. So as I think everybody in this room is aware, our Pacific salmon return along the northwest coast of North America in the fall every year in order to spawn in their beautiful fall regalia, and that after they spawn, they die and return the nutrients that they accumulated in the ocean to the freshwater ecosystems where their offspring are rearing. And so this is how things are supposed to work. In the early 2000s, we started to document this phenomenon of fish that were returning to urban-impacted creeks. As we now know, it's more roadway-impacted creeks to spawn, and were dying before they got a chance to spawn. And these fish, mostly look in perfect health. Some, like this example here, still haven't even become fully sexually mature, recently returned from the ocean. One of the questions I get most frequently over the years as I've been talking about this is, well, what happened in the early 2000s that coho started dying in urban impacted creeks? And the truth is that they didn't start dying in the early 2000s. We started to notice a problem that had been going on for decades. And how did we start to notice this problem? Really, ultimately, we started looking in the right place at the right time. The serendipity of this discovery is actually something I wanted to point out today. So we had to be looking in the right place at the right time. And the reason that we started to notice ultimately is tied to the fact that in the late 1990s, early 2000s, we started spending millions of dollars to restore urban impacted creek systems. And the part of the reason for that is that salmon in the United States had now been listed, various species have been listed under the Endangered Species Act, right? So suddenly there was an influx of dollars and attention being paid to the fact that we needed to restore these urban creeks to help restore salmon populations, right? To make that effort. Other things, though, that were also required for us to notice this problem. Oh, I meant to say, this was actually my first job, so it's interesting with the stream restoration. This is like working on the riparian system to make sure there are native plants there, working on, installing engineered large woody debris and gravels and boulders to get the physical habitat attended to. And this didn't, in fact, address the quality of the water that was entering these systems. And in my firsthand knowledge on this was in fact my first job when I moved to the United States from Canada in 1998, was working on stream restoration as part of those initial efforts. Some other things that were required for us to notice this problem was a presence of coho salmon that would be using these systems, right? So we had to have restoration in systems that were going to have this impacted water quality. And the restoration allowed for there to be the potential for spawning and rearing in these systems. But we had to have the fish. The reason that we had fish in these quite impacted systems where water quality is pretty strongly impacted is very likely because of net pens that were operating out in Elliott Bay, right outside of where a lot of these urban creeks are, that where the tribes were rearing coho salmon since 1993. They still do this today. And in the fall, these adult fish are now looking for somewhere to go, right? And here's some restored creeks. They're like, cool, I'll just head up here, right? So that's one thing. Another thing, abundance of fish or presence of fish. Post-restoration monitoring. A lot of these projects didn't do that. A lot of the restoration projects did the work and then walked away, like, whoo, we did it. But some, including the one that ultimately led to the very first observation that was reported to Fish and Wildlife Service, was looking in the fall to see if adult salmon had come back to use these systems. The final thing that was required for us to understand that there was a problem is actually catching the fish at the time that they were sick. Observing the fish while they were sick. The mere presence of dead salmon in a stream in the fall does not indicate a problem, right? Because they're semelparous, they spawn and then die. So it really required seeing those fish sick to notice that something wasn't right, and then opening the fish up to see that they had not in fact spawned. So we started doing that. And here's just another history picture of me. In 2004, I was the first coordinator of pre-spawning mortality studies, as we called it at the time, where I organized crews of volunteers from various institutions to try to understand the extent of the problem out in the field. And in fact, across ultimately 7 years of field studies, we were able to demonstrate that this problem was widespread, it was recurrent, and here's that example of, the female opened up clearly unspawned. We know that this also happens in male coho salmon, but it's a little harder to tell if they've already partially successfully spawned, right? Easier to tell with the females. So we were able to summarize this research ultimately in this 2011 publication and showed that the weight of evidence, I'm like, what are the things I say here? Right. The weight of evidence pointed towards stormwater runoff because common conditions or common problems in a watershed that might cause pre-spawn mortality were not present here. There was no elevated temperature problems, no low dissolved oxygen, no evidence of like a big parasite infection that would have led to mortality. And we couldn't explain the mortality by any single contaminant that we knew of. There was evidence of exposure to metals through accumulation in the gills. There was evidence of exposure to hydrocarbon accumulated in the bile processed through the liver, but those could not explain, based on what we knew of the science, the mortality that we were seeing. That's that slide there. Okay, so pointed towards stormwater runoff, right? And what we know about the natural environment is that most of the water that falls that doesn't evaporate actually infiltrates into the ground, right? Very little of it runs off as surface runoff into receiving waters. Whereas in our built-up environment, most of our surfaces end up being impervious, right? So we've got our roofs and our driveways and our roads and parking lots and buildings, and most of those don't allow water to infiltrate into the ground. Instead, as that runoff flows off of the surfaces, it carries a ton of pollutants into the receiving waters. Think about not just what would be on roads and horizontal surfaces. Should I admit them, or is somebody else doing this? Here, there. But even just the fact that you would pull contaminants, we pull contaminants out of the sky with rainwater falling, right? So particles that are in the air will fall out. We can leach materials out of our building materials. Leach chemicals out of our building materials, pick things up off of the horizontal surfaces, and they all flow into the water. This is a cool example if you haven't seen this video before. It's a time lapse of an outfall where stormwater is entering Puget Sound just off of Alki Beach. And what we're seeing here is the accumulation of all the different types of runoff that end up getting shunted into receiving waters. And we're seeing the particles that are carried by that runoff, but we're not seeing the dissolved chemicals, which as a toxicologist is the part of that profile that I'm concerned about. This video actually goes through the night, just as a powerful example of what it looks like when that water is entering, the runoff is entering the receiving waters. Along the way, one of the clues we had that roads might be really important to this problem is this study. I was not directly involved in this study, but it's a really important piece of the story. So by surveying, I think there were 21 basins throughout the Puget Sound where there was data done by various groups of the rate of pre-spawning mortality of coho, associating that with the land uses in those basins and doing fancy statistics, they were able to show that it wasn't just impervious area generally that was correlated with rates of mortality, but much more specifically, the amount of roadways in a sub-basin was correlated with the rate of mortality in that basin. So pointing towards something coming off of roads, and the busier roads had the higher correlation with mortality. So maybe not even roads, but traffic on roads, right? So this all led to collecting runoff and actually starting to test what is the runoff doing? Is this really the source of the problem? And yeah, it was. This is just a historical photo at this point. The first runoff that I ever collected in 2011, and the downspout it came from. I was working as a postdoc at the time at Washington State with John Stark in a project in collaboration with Nat Schultz at NOAA Fisheries and Jay Davis at U.S. Fish and Wildlife Service, and that's what started our understanding of what the runoff's really doing to our salmon. But we experimentally exposed coho salmon to the runoff that we've collected directly from a roadway. And each of these PVC tubes contains one adult salmon. And if you haven't done this kind of work or seen this kind of work, it's actually a good, safe way to work with adult salmon who are otherwise wild, powerful wild animals, right? So in doing this, we were able to actually show that even brief exposures to runoff were able to recreate the symptoms we were seeing in those urban creeks and the mortality that we were seeing. And we were also able to show through various studies over the years that this affected not just the adult spawners, but also the alevin and the juveniles. We learned that it didn't take very much of the runoff to cause this problem. This is 3 different storms that we collected runoff from, showing that at 25% runoff, we have pretty much 100% mortality, and some mortality occurring even down at 5% runoff. And early on, Ed's best guess based on various non-target chemicals present in runoff and then present in creeks said, well, you could certainly have about at least 15% of your creek water could be something as strong as runoff from this highway, helping to explain the mortality we were seeing. So not just low concentrations, but also short durations we found of exposure to runoff could cause mortality. We're seeing here the mortality in the black bars, that's mortality that took place during that exposure duration I'm showing, and then the rest of the time was in clean water for those shorter than 24 hours. The black bars are for fish that died during the actual exposure, and the white bars are fish that died during the rest of the 24 hours in clean water. So this showed us that it doesn't have to take a very long pulse of runoff into the receiving environment in order to cause mortality that might take place the next day. Okay, by using juveniles, we were able to study the progression of this illness. One of the first things that we noticed by recording, because the runoff is dark enough, we were recording video at the surface of the water, and we were able to see that. So this is runoff that was causing mortality by like 6 hours. Within the first hour, we were able to see a behavioral change away from controls, where they had an increased rate of discrete surfacing activity within that first hour. That then progressed into continuous swimming at the surface, which progressed into losing equilibrium and ultimately laying on the bottom of the tank before dying. Importantly, from this study, we found that once they'd reached that continuous surfacing behavior, they would not recover. When we transferred them into clean water at that time, they died in the same timing and the same rate as the fish that just stayed in runoff the whole time. So that lets us know that the body burden that they accumulate to cause that mortality occurs pretty early on. We didn't actually test fish at different stages here. This was just like, oh, let's take these continuous surfacing fish and put them in clean water. Other things we learned along the way about what's happening as they get sick. We see changes in their blood chemistry, so osmoregulatory imbalance and as a reduced sodium and chloride concentrations, increased pH. Those, however, look like they could just be part of a general stress response. We also see hemoconcentrations. The blood gets really thick. And this is well beyond a stress response. Blood can be about, it's as much as twice as thick as the healthy fish. So we wanted to learn more about that, what was going on with that, because that increased hematocrit was much higher even than fish that died from extreme stress. Sorry, what I mean is exercise stress. You can swim an animal at the fastest rate possible until they die, and increased blood thickening, increased hematocrit is part of what you see changing in those fish, but the levels that we see for fish exposed to runoff are way beyond even that. So we were looking into this. My PhD student, Nagone Blair, was sampling fish exposed to runoff by the time they lost equilibrium, and then injecting into their bloodstream Evans blue dye. And this is a high molecular weight molecule that will fluoresce under certain light conditions. So allowed it to circulate through the vascular system of the fish, and then perfuse the fish with saline, so just basically flushing out the blood with clean water. And here you can see their gills are not the red they're supposed to be because the blood isn't there anymore. And then what we did is measured the amount of dye that was present that had remained in the fish's bodies. And this should only happen where plasma, the watery part of the blood, and molecules large enough, like this Evans blue dye, had leaked out of the vasculature into the surrounding tissues. And fish and us as well, we have vascular systems that are leaky to various extents. Certain organs are intentionally leaky, like our gut, right, to allow things to go from the blood into our bloodstream. But some places we should never have leaking. And so we were particularly concerned when we saw leaking in the brain region of these fish exposed to runoff. That shouldn't happen. The blood-brain barrier should protect the fish from that happening. So a little bit about that. Capillaries are our smallest blood vessels, right? And the tube of the capillary is actually these endothelial cells that are linked together really tightly. And they're kind of stapled or glued together by these tight junction proteins so that there aren't any gaps in them. Now, leaking that does take place could be a result of damage to the actual cells themselves, or molecular signaling can tell those proteins to let go and cause a gap that way. So we wanted to learn more about the timing and the severity of this leaking that was taking place. So we again sampled fish at the loss of equilibrium, but also 2 earlier time points. So once they'd reached that continuous surface swimming, and also as pre-symptomatic. And we quantified not just the high molecular weight, the large molecule tracer, but also a low molecular weight, smaller molecule, sodium fluorescein. So we thought, well, maybe initially there's like a little gap and little molecules get out. But what we ended up seeing is that both the high and low molecular weight tracers were present in all of the fish that were showing these advanced symptoms. And in fact, some of the pre-symptomatic fish, you can see like 2 of those fish also had leaking before they even developed any of these obvious symptoms. So this lets us know that the leaking of the vasculature in the blood-brain barrier in this particular case is necessary, if not also sufficient, to cause those behavioral changes. We have now done this experiment also just with 6-PPD quinone and see the same results, and we're also able to demonstrate that there's leaking of the blood-gill barrier as well. So it's not limited to just the blood-brain barrier. It's probably taking place in all the vascular tissues. One more thing here about this study. We're just getting ready to publish this, so I'm all excited about it. We wanted to actually look at the brain tissues and see where this leaking was taking place. Would it be in certain parts of the brain? This is what we expected to see, is that we might see leaking around the capillaries. This is called punctate staining. So we injected horseradish peroxidase, which with DAB and hydrogen peroxide will turn this dark brown color wherever it is in a tissue, and this should be where it has leaked into the brain. But this is not what we saw. Instead, we saw this, which demonstrates that the leaking is pervasive, and it's also very severe. Okay, oh, and then there was, one more thing here. So in the runoff exposed fish, we did see some stenosis where the blood vessels get constricted and they actually are trapping red blood vessels sometimes. But on the right image here with those arrows, what we're highlighting mostly is looking at the actual endothelial cell that makes the capillaries. And we're able to see from this that the endothelial cells were not damaged. So it's not that they're dying and breaking apart and that's what's causing the leaking. Instead, it looks like it is some sort of signaling causing the blood-brain barrier to open. Okay, other things we learned along the way is that coho that sensitivity varies among species of salmon, right? So in our region, coho salmon, probably here too, coho salmon return and chum salmon all will return at a similar to overlapping time. So we were able to co-expose those guys and see that the chum salmon not only did not die from runoff exposure, didn't show any changes in their behavior or their blood chemistry. And this agrees with field observations we had, where this is a coho who is expiring in the creek, and the camera will pan up to show a very happily, robustly spawning pair of chum salmon. And in our field surveys, we wouldn't see female chum salmon full of eggs, so we were pretty sure that they were unaffected from that perspective too. All right. By testing juvenile salmon, we were able to see that there was a gradient in sensitivity among our salmonid species, the ones we've tested so far. The next most sensitive from coho was a steelhead, juvenile steelhead we tested, and some of the juvenile Chinook that we tested also died, although we know now that they're much less sensitive to 6 PPD quinones. So there might be a mixture thing going on. In contrast, sockeye salmon, sockeye salmon, chum salmon, and we think also pink salmon are basically appear insensitive to this acute lethal effect. All right, so what is in stormwater that makes them sick? Well, we've obviously learned a lot about that, but there's a little bit of that history. Obviously, the chemistry of stormwater runoff is super complex. You know, back in the day, we're like, oh, we know what's in runoff. You know, there's metals, there's hydrocarbons, there's some pesticides, blah, blah, blah. You know, we even the hubris, we even thought, you know, we could just make stormwater and you know, study the problem that way. Bowen Du was a postdoc working with Ed Kaloje and was able to you know, wake us up a little bit. There were actually thousands of unique chemicals present in roadway runoff. Kathy Peter also worked with this and began identifying some of those chemicals. Lots of them were just unidentifiable. And even the ones we knew about, we didn't necessarily know much about or anything about their toxicology. And finally, the chemicals that we did know something about, you know, those metals and hydrocarbons in particular, also pesticides, didn't appear to be able to cause this effect at the concentrations that they're present at in the field. We actually made mixtures of metals and hydrocarbons in this Sprongberg et al. Paper, even tenfold higher with the metal concentrations, and nothing. So clearly we were missing something, and rather than sort through that chemical soup, chemical by chemical, we asked the question of, well, what are the sources of toxicity, sort of the sources of chemicals to roadway runoff? They're coming mostly from our vehicles, we reasoned. We have particles coming from tire wear, from exhaust, from brakes. We have liquids that we either intentionally release, like windshield washer fluid, or that leak out of our vehicles, right? So each of these sources is itself a complex chemical mixture. So we reasoned that if we could identify one of these sources as maybe the source of the problem, we'd have a much smaller haystack to look through. And one clue to focusing on tires came from this work in collaboration, but led by UW-Tacoma, where we're looking at various of those sources. We're developing these fingerprints for the chemicals that are present there. And this is, I think, was like 58 chemicals that were often in waters that killed coho salmon. So you can think of these vertical columns as individual chemicals, ranging in abundance from not there in the dark blue to highly abundant in the dark red. And then comparing those fingerprints essentially with fingerprints for roadway runoff samples that we know killed coho, and receiving water samples where coho have died around the timing that the water sample was collected. And then sorting these in terms of chemical similarity, identified just one source, tire particles, that looked like it was very chemically similar, at least more than these other sources, to the waters that kill coho salmon. So an interesting thing about this, right, is that the presence of a lot of tire chemicals in water didn't have to mean that it was a tire chemical causing the mortality. So we did have to go back and ask the fish what they thought of this, creating a mixture of tire particles from the tread of various tires, you know, trying to cover a range of attributes, but not trying to hit everything. Grinding them up, passing water over them for 24 hours, just ambient temperatures, to see what would leach into the water, because that's probably what's leaching in the water in the field. And then we asked the fish, okay, if tires are an important source of toxicity, would they respond the same? So if they did, we would see acute mortality in coho from exposure to the tire leachate. We would not see that response in chum, right? And we would see that the coho got sick in a similar way from exposure to runoff and from tire leachate. And over a series of experiments, we did in fact show that yes, coho would die when exposed to tire leachate. They died in the same way as when we exposed them to stormwater runoff, and the chum were not affected. So from here we went to trying to identify the chemical in the tire mixture that was responsible. And to do this, we paired toxicology exposures with high-resolution mass spec. We created that tire leachate, and then we started to reduce the chemical complexity by fractionation. I don't know much about this, so I just use the first couple examples of, for example, if the particle was attached, if the contaminant was attached to a particle, putting it through a sand column should filter out the particles, and what came out would be clean. So we did this fractionation and would generate a toxic fraction and up to several non-toxic fractions that we would then test on the fish, using them as to trace the toxicity, basically. If it killed coho, put it back on the machine, try to understand the chemicals that were there, and at the same time continue to fractionate and reduce complexity. Now this approach worked, as you know, but when you think about it, it's a little scary to think it didn't have to work. Like, we could still be floundering around. This method does not capture all chemicals. It might have required a combination of chemicals that got separated in the fractionation process. It's a little nightmarish to think about the ways in which it might not have worked, but it did work. So we started with this mixture that contained over 2,000 chemicals, generating one toxic and several non-toxic fractions, ultimately ending up with a mixture that contained just 4 chemicals dominated by the C18, H22, and 2O2, which we didn't know what it was. Zhenyu Tian, who's the lead author in the paper and was a postdoc with Ed at the time, he was the one that made the association between this unknown chemical, and NMR was able to tell us what it looked like, but we still didn't know what it was. Not present in any databases, not a known tire ingredient, but Zhenyu made the association with this 6-PPD chemical that is an ingredient in tires, same number of carbons and nitrogens as are unknown. They then purchased 6-PPD industrial grade, and through ozonating it, were able to generate, among other transformation products, our unknown that we then called 6-PPD quinone. We then tested that against our mixtures, against this 6-PPD quinone isolated from our ozonated mixtures versus, and also isolated from our tire particle leachate, found that while the parent chemical 6-PPD could cause mortality in our coho salmon, it was this transformation product that was super acutely lethal. All right, so confirming for us that this is our toxicant. We also looked at the 6-PPD quinone in those environmental mixtures. So the top graph is, well, they're both showing us the percent mortality of coho salmon in a 24-hour exposure, exposed to dilutions of either, in the top panel, we have stormwater runoff and we have tire leachate, so the mixtures containing 6-PPD quinone. And the bottom is just pure 6-PPD quinone. And that red dashed line indicates the concentration that killed 50% of the fish. That's the median lethal concentration, or the LC50. So this allowed us to see that, because those LC50s were pretty much the same number, that the toxicity of those the complex mixtures containing 6Q, sometimes I abbreviate it too, could pretty much be explained by just the 6Q without having to consider the thousands of other chemicals that were also present there. So our primary causal toxicant is how we decided to talk about that, because we're like, there could be other things contributing, right? But it looks pretty explanatory. We also at the time were able to show that in our roadway runoff samples, this chemical was present above that LC50, above the median lethal concentration, so you would expect to see mortality. And also in stream samples and waters from not just the Puget Sound, it was also present at sufficient levels to cause mortality. So we reasoned that you would expect to find this chemical anywhere where tire particles were getting into the water. And in the past 4 years, obviously research from other areas have confirmed this, showing that around the world, we find 6-PPD quinone not just in water, but in air, in dust, in snow, everywhere that there are tires. Our environmental relevance obviously comes from the fact that 6-PPD is the primary anti-ozonant that's used in tires, and it's necessary that it's there to protect against the ozone. Ozone will interact with the surface of the tire. It can penetrate up to like 20 microns into that surface, and it's there to, 6-PPD is there to scavenge the ozone and prevent it from breaking the tire polymers. As those breaks get deeper and deeper, the cracks extend to the point where you run the risk of compromising the tire while it's in use, which is very dangerous, right? So there's enough 6-PPD in a tire to migrate slowly to the surface over the life of the tire, preventing this problem largely from happening. And if you haven't thought of this before, the brownish color that tires can get is because of 6-PPD quinone. Don't buy the products that allow your tire, wash the bloom off your tire and make it all shiny and black, because this actually prevents further oxidation from happening. So this chemical is more soluble than its parent as well, so we know that it will leach directly from the tires or from those warm particles, and that is how it gets into receiving waters, other ways too. Okay, other things we've learned along the way, other species of salmonid. I'm just showing the salmonids here because the list is getting so long for species that have been tested, that we have some other salmonid species that are sensitive as well. And then various other salmonid species, even in our region and closely related to coho salmon, that are not sensitive. One of the things we wanted to do for our follow-up paper where we revised our LC50 was just to demonstrate how very toxic this chemical is. So I pulled 4 chemicals that have an aquatic life criterion. These are concentration limits that are meant to protect against that chemical getting into water. So for the most sensitive chemicals, the most potent chemicals with the aquatic life criterion that had the lowest values, I pulled the data for the most sensitive species that have been tested to develop those criteria. And these are the most sensitive of those up here. And 6-PPD-quinone falls right at the top of that list. And depending if Bonnie Lowe, I think, is here, we have a number that matches that parathion number. So this indicates to us that it is among the most toxic chemicals that we know of for aquatic life, which is kind of amazing, right? And that it's up here with organochlorine and organophosphate pesticides mostly that are neurotoxic, are designed to be acutely lethal. Okay, so there has been a very rapid regulatory response, right, to this issue, which is great. And I wanted to say a little bit about the context of that, right? Salmon species are protected. There are legal protections for these species in North America. This map here is just showing us that there are a variety of regions, this is for the US, where species are listed under the Endangered Species Act. And this comes along with requirements and opportunities, funding support, to try to preserve those species and dare say, recover those species. We also have federal obligations to uphold tribal treaty rights, right? In our bioregion, the tribes ceded their lands in the 1850s with the understanding that they would have the right to hunt, fish, and gather in their usual and accustomed areas. And that is clearly being threatened by a variety of things, including water pollution. And these rights are, they're a treaty between a sovereign nation and the federal government. So they're not just nice to have, they are legally binding, and that does hold certain protections. Finally, we have the Clean Water Act in the United States that should protect against toxic pollutants and toxic amounts. And this underpins the state water quality standards that include the aquatic life criteria. So we are proud that we do actually have an aquatic life criterion that has been developed, and that this is a good first step, right? But we have a long, long way to go. I like to say that we have a long road ahead, and it is still filled with tires, right? So what are we going to do to try to meet these limits and not be in violation of the Clean Water Act, which obviously at these numbers, most creeks that aren't even heavily impacted would exceed these numbers. All right, so some of the ongoing solutions, marine stormwater infrastructure, and I've seen a bunch of posters. I know some of you publish in this area too. At the Washington Stormwater Center, which is a collaboration between Washington State University and the University of Washington, we study a variety of these technologies that are meant to encourage water to spread out and soak in instead of just running into a surface water body. And what we spent a lot of time studying has been bioretention. And in Washington State, this is a pretty simple mixture of sand and compost, 18-inch depth, I believe. And well before we knew about tire chemicals and the problem that they were actually causing, we were testing whether this technology, this approach would prevent the toxicity. So here we have fish exposed at 4 hours into a 24-hour exposure to either the well water, the unfiltered runoff, or on the far right there, the same water passed through that bioretention system. I may have to click it to make it work. There we go. Completely preventing the mortality. And we've seen this not just in adult spawners, but also in juveniles and alevin. So whatever it is, we know how it works, right? We don't know how it works. We know that it works. And to the extent that we can spend more effort really understanding these systems, we have the potential to optimize them and be more effective in their placement on the landscape. We just finished this study in which we collected roadway runoff over 4 years and applied it at an accelerated rate. This happens to be 3 depths of bioretention we were testing, but all 3 depths responded similarly in that they were able to continue to prevent the coho mortalities through the entire 13 water years of the study. So applied the runoff at an accelerated rate that was ultimately 13 water years. Still, as a researcher, you're hoping to show like, oh, this eventually, stops working, and this is the timeline at which we need to replace it. We were hoping for a different result, but this is actually great news for our installations. We've also looked at some other systems, so bioretention containing inoculated with fungi and containing plants. And these showed pretty much 100% removal in the study that we were doing. We have also looked at compost-amended bioswales, which do more of a horizontal flow treatment, and those were also fairly effective at removing 6-PPD quinone. So we have the opportunity to install green infrastructure to deal with this problem, but there's a lot of limitations with that for places that are already built up, right? Like Seattle, for example. So in addition to making sure we get green infrastructure on the land where we can, and then trying to learn more about how it works so we can optimize it, we also need to be focusing on source controls. And we've been in conversation with tire manufacturers through the USTMA since before we actually made the discovery. And they were quick to respond and to understand, I think, that we need a solution here. So in our lab, as part of that, we have been studying the relative toxicity of potential alternative anti-ozonants to replace 6-PPD as a safer replacement product. And Caitlin Lawrence had led this work when she was a master's student. So this is just showing that relative mortality across a 24-hour exposure, which is sort of a forced period of time, right? Much more important than this is the ozonated mixtures that result when these parent compounds get exposed to ozone. And Anya McConaughey's here and has a poster on that out here because she is continuing that work for us. All right, the last thing I wanted to do was just highlight the many organizations involved in this research over the past 20 plus years, and even more specifically, funders involved over the past 20 years. And I put the US EPA logo as really big because in our current political climate, obviously there's a lot of concern about losing these sources of funding. And I put that really large because over the 20 plus years, the US EPA did provide the majority of the funds, either directly or indirectly as pass-through funds, to be able to address this problem and get us where we are today. And so to the extent possible, we would like to make sure that this agency and other federal agencies continue to be able to provide this support. And very finally, I want to acknowledge the coho salmon that have lost their lives in our efforts to try to understand this problem, and the coho salmon that are have lost and are continuing to lose their future generations to this problem as we work towards a solution. Thank you.